Hello guys, welcome to the Inspiring Minds coaching, and now this is the second part of the bacteria structure. As in my previous video I told you about the essential part of the bacteria. They were including the nucleus, cytoplasmic materials and also the cell membrane, or you can say the cell wall. So here we are going to talk about the non-essential part of the bacteria. See, make it clear, because in the previous video I also told you that non-essential part of the bacteria, that doesn't mean it is useless. It is very essential part for the diagnostic point of view or the causing disease. Okay, so let's begin. As our heading says that it is a non-essential part of the structure of the bacteria, and I have given it a special point as a pathogenic matter, or you can say the pathogenic material. Almost every kind of infection which is caused by the bacteria have been identified. They are essentially the trigger point and initial portal or path belong from the non-essential part of the structure of the bacteria. This is a complete content where we study about their physiological component of the bacteria and also their pathological. So let's see one by one. The very famous, the first one, capsule. From the first video I have told you that like this is the bacteria, and here you can see if it is nucleus, and here extraplasmic plasmid cytoplasmic granule, and there is a thick cell wall. In my previous video I told you about the cell wall component, and there is pili, and there is membrane, and just outermost layer which is capsule. Remember, in my second last video I told you about one of the very important property of the capsule, that it protects bacteria itself. Okay, if you see here and you may ask that protect bacteria and it is not essential part. See why it is needed. When some kinds of activity happen, then only we need protection. If we are generally living in normal society, so we don't need any kind of police or watchman, someone to protect us. But when there is any kinds of attack, or it may be thief, when they are, so we need a kind of protection. There should be any kind of policeman, guards and watchmen. So they need only a condition when there is harm, or you can say there is a danger to the bacteria itself. Otherwise it's not needed. But you can understand that bacteria is going to cause a disease, and obviously if you slap someone, then definitely he will react or she will react. So that moment you should have a defense mechanism. This capsule work as similar as that defense mechanism. Okay, so here capsule is composed of a polysaccharide. Polysaccharide in the biochemistry, Dr. Rishabh have told you about that. Just I will give you the briefly that polysaccharide is carbohydrate where carbon, nitrogen, hydrogen and oxygens are in specific combinations, and they are found almost all kinds of living organisms on the earth. Their component is polysaccharide. And from the exam point of view, you need to remember that only this word, that polysaccharide. You don't have to be confused. A capsule is composed of, like if someone say lipid, no, or someone say protein, no, they are the polysaccharide membrane. And what is their function? To the function point of view, the capsule protect bacteria against, that is function, and that is component. See, I have given only a one word answer so that you can solve the maximum question, and they are composed of, component is polysaccharide and their function to protect bacteria against phagocyte. What does it mean, phagocyte? In the pathology and also in medical biology, you have learned about the pathogenesis. Pathology, pathogenesis, pathogen, all these things, names itself have says that there is a pathological or disease causing condition. So here is a phagocytic activity. So you can understand that protect against phagocyte. Who caused the phagocyte? Our body have a system, body fluid system, which is very famous for us, that is known as blood. In the blood, what we have? There is blood, and in blood there is RBCs and WBC. WBC have agranular and granular. Granular have basophils, eosinophils and neutrophils, and agranulars have the monocytes and lymphocytes. Monocytes further form macrophage of different organ like liver, Kupffer cell, in lungs, dust cells. These all things comes under pathology, and in the pathology we will study about those macrophages. So what happened? Those macrophages forms when there is antigenic stimulation in our body. When bacteria enter in our body, so what happened? They have a special type of interaction with us. It may be injury through our food, or it may be some kinds of infection that is unwillingly we have received. So what happened that moment? That capsule is just found the outer membrane layer. Okay. There is some kinds of secretion, and also that is a bacterial additional properties. So what they do in our blood, there is antigen specific. In our blood, there is antigen specific cells. They are neutrophils in combination with the macrophages, with the lymphocytes. They perform an apparatus work as eosinophils. Okay, they perform phagocytosis. Phagocytosis. Okay. Phagocytosis, and that activity, when we talk about specific, that is phagocyte. So what are those phagocytes? Now you can understand. Phagocytes are those cells which work as to kill that microorganisms or any kind of pathogen containing organisms which enter in our body. Phagocytes are specialized cell. They may be modified from the monocytes, or they may be modified from the lymphocytes. So you have to keep it clear with your mind that phagocyte is just to diminish the phagocytic activity of those microorganisms for which you are talking. So make it clear that when you solve the question, so you can't say that capsule act as is its function to protect from the macrophages. No, because all are combinedly acting over the in this system. Just phagocyte is the unique and confirmed term. Okay, so in this case you can see that the protection against the phagocyte. Why? So that disease will not occur. Their property to protect against phagocyte. So what phagocyte do? Phagocyte if present, like, okay, you can understand like this way. If phagocyte present, so disease will not happen. But if phagocyte absent or phagocyte is unable to cause its activity, then there will be disease. If it is absent, then there will be disease. Okay. Each and every time whenever we talk about the phagocytic activity or the bacterial structure and function, when you can talk, you can see there is exotoxin, endotoxin, and lots of disease. So there you have to keep it clear in your mind that it may be caused by the any kind of secretion or any kind of component which belongs from the bacteria, but most of the time, I can guarantee you that is 99% time you will listen, or you have to answer only and only one word answer, that is capsule. At the broad level of infection of the bacteria caused with a capsule. And the second bit, and here the second one, which is the non-essential part of the bacteria, that is pilus or fimbriae. Remember in my first video I told you about that pilus is represented as Pi and fimbriae as F. Pi and F. That is F and that is Pi. This is composed of glycoprotein. If you study clearly biochemistry, then I think you know the polysaccharide and glycoprotein's difference. But if you don't know, don't worry. The glycogen containing protein, glycoprotein. Simple. Okay? Glycoprotein form pilus and fimbriae. See what happened, that here is the cell wall and innermost plasma membrane, and here is the capsule, and there is outermost there is fimbriae, and there is a small structure, that is pili. Remember pili like structures when we talk about the epithelial cells in animal cells also. So similarly there is also living organisms and pili is present. Many times when you listen the word that is pili, it means a small finger like structure, the very famous line of the NCERT. They are the glycoproteinaceous component found on the bacterial outer surface, and they are non-essential structure for the bacteria itself, because they are not important, not necessary for the bacteria itself, but they are very important and very dangerous, because they cause disease and they help them to escape before diagnosis. This one protected from the phagocyte cells which our body have. That may be systemic circulation, or you can say there is a lymphatic circulation, and the more accurately there is natural killer cells, T lymphocytes and macrophages, and a specific apparatus is formed which we study in the pathology and biochemistry, and their phagocytic activity have been diminished because of the capsule. Okay, and next one, pili. What happened, that they are a small structure, and what they do, very important, their additional function is locomotion and movement. Locomotion and movement. Okay, what is the difference between locomotion and movement? Like I am standing here, okay, so I am using my hand, that is my movement. I am using my neck, that is my movement. But when I move like this or like this, that is my locomotion. Okay, I use my limb and I go from here to there, that is the locomotion example. When I talk about even our eye blink, that is a kind of movement, but it is not a kind of locomotion. Simply keep in mind, locomotion means you are going to locate from one place to another place. So here the longer part is fimbriae. It was very important question in NEET UG, and many time in NEET PG it has been asked application based on the fimbriae. They are the glycoproteinaceous. There are basically two types. What are those types of the fimbriae or the pili? They are pili, and what are those two types? There are two types containing, first one pili and another one fimbriae. Their names are interchangeable because pili and fimbriae also, what they do, they help in attachment. What does it mean, attachment? Like here is your gut, okay, and here is intestine, and here is pancreas. If bacteria enters from the oral route when you eat something very bad or bacteria containing or contagious food, and they should have ability to stick over the surface to cause the infection. At the basic histological level, what happened that those bacteria stick over the surface, they release some kinds of chemical substances which is known as toxic product or toxins, and they enter in our lymphatic or the systemic circulation, so that they further go to the other part of the body. And if our body have the strong capability which is known as the defense mechanism or immune system, they protect from the pathogenic activity and they kill or destroy that pathogen at that moment. But if our body is unable, so that bacteria or that pathogen cause disease successfully. So the pili is very important and fimbriae for the attachment. Remember, phagocytosis, there is 5 steps. The first one is attachment. So it should be there. So you can understand it in this way, that pili and fimbriae are the very important for the bacteria to land over the surface of the any macroorganisms, or you can say the host. So you can here mention one more thing important for the host attachment and survival. What does it mean, survival? For survival, we eat, we breathe, we eat, we breathe, we move, we digest, we excrete. But one thing more, if you want a generation to carry another generation, so there should be a reproduction. In my previous video, I have told you about that bacteria, most of the time they have done the binary fission, but very important to keep in mind that there also I said not only binary fission possible in bacteria, because other reproductive mode or other reproductive paths are also available in the bacteria, which is known as conjugation. So just quickly make it binary fission. What you have learned. I will again, when I will talk about the bacterial genetics, then I will tell. And conjugation, binary fission, transformation, these are the reproductive part of the bacteria. And here the next one, that is flagellum. When we talk about the flagellum, so don't make it mix with the fimbriae, because it is FL and it is FI. So here fimbriae is used for the attachment, and here flagellum is a protein containing small structure. They help with the movement of the bacteria. Movement of bacteria. Okay. And now the next one, biotechnological or genetic point of view, plasmid. Plasmid is very important for the bacterial inheritance or bacterial genetics point of view, but plasmids have almost no any kinds of use when we talk about any kinds of infection or pathogenic activity of the bacteria. What happened that from the very beginning you have learned that plasmid is the extra chromosomal material found in the bacteria. There is DNA in compressed way. So simply what happened, that DNA is present in a specific form, like you can say like this way, and their DNA is present, and a small glycoprotein layer is there. So they may contain that positive and negative, that is gene containing. Okay, there are two very important functions of the plasmid which have been asked in the question, that they carry antibiotic resistance or antibiotic resistance gene, and they are able to produce toxin. All three options were right when you solve the question in a FMG. So I have mentioned today the capsule, pilus and ephemerae, flagellum and plasmid of the non-essential part of the bacteria. Keep in mind each and everything where I have mentioned the star. It is very important for the question point of view. Just don't make it so much confusing for you. This was just to understand this. Okay. For FMG and NEET PG examination or IFOM examination, IFM always ask about the capsule and plasmid. Capsule and plasmid. From IFM point of view, every time you will see the question which have been asked from the capsule and plasmid. Just capsule, all about capsule, their activities, their functional property, and plasmid. So you don't need to be confused. And capsule structure, and because of the capsule staining, many times what happened that we are able to identify the clinical things, or that is known as clinical diagnosis. In NEET and FMG questions, most of the time they have asked the plasmid character, antibiotic's name, and sometimes conjugation history. Okay. So in the next video, I will tell about few more structure of the bacteria, non-essential part of the bacterial structure, and then we will move to the question of all those questions which have been asked on the behalf of these structures. Okay. So hope you guys like this video. Please like, share, and subscribe. That's Spine Mindz Coaching. And if you have any doubt, anytime you can comment or you can ask to our representatives, and they will definitely able to tell your questions to us and we will answer. Okay. So here is everything is clear. I think if you need anything for me, anything to make it clear conceptually, because it belongs from the histology and pathology I told you, and this one from the bacteriology, but in combinedly all these things will let you solve the questions. If you know only the bacteriology, don't know basic concepts, you are unable to solve the questions. Okay. So let's meet in the next video. Thank you.